Resuming our discussion on oxidative phosphorylation.
This slide highlights a lot of what goes on in oxidative phosphorylation, and we'll skip over the glycolysis, the citric acid cycle, and we'll leap right to NADH, which is the high energy electron carrier, donating electrons in succession to 3 different protein complexes. Each of these protein complexes will be pumping protons or hydrogen ions from the inner mitochondrial space to the inner membrane space. And finally, the electrons will be donated to oxygen to form water. All the excess protons that exist in the inner membrane space will then be able to diffuse down their electrochemical gradient through the ATP synthase, a protein complex that's also called the F1F0 ATPase. As the protons go down their gradient, the energy will be captured in part and used to join ADP and inorganic phosphate to produce ATP, which is as you know, the high energy molecule which drives a lot of unfavorable chemical reactions. Now, one last comment here is this slide shows that physically the electrons are moving completely flat in the membrane, but they're going energetically downhill. Highest energy electrons, medium energy electrons, and lowest energy electrons. And for all that activity, this slide summarizes just the chemical changes, and that is NADH gets converted to NAD+, oxygen and protons are converted to water, and ADP and inorganic phosphate is converted to ATP. The next video will talk about in detail the mechanisms that occurs, but these are the net changes that have occurred. Now this slide focuses in detail on the inner mitochondrial membrane here, and we'll use it to talk about the coupled vectorial processes that occur. The first process is electron transport, and the slide has changed direction here. In this case, the electrons are being donated. The first protein complex, which happens to be NADH dehydrogenase complex, is on the left. In the previous slide, it was on the right. Anyway, the high energy electrons get donated to this protein complex, and we're transferring electrons within the membrane laterally from the high energy protein complex to the medium energy protein complex to the lower energy protein complex. And this process drives another vectorial process, which is the pumping of protons or hydrogen ions from the inner mitochondrial membrane matrix to the inner membrane space. The excess protons are free to diffuse in this space. Protons will diffuse down their electrochemical gradient through a protein called the ATP synthase, and that's what's driving the synthesis of ATP. So the coupled vectorial processes are electron transport, proton pumping, proton diffusion. And it's interesting to think about the different ways that the cell has maintained high energy currency. It starts with the high energy sugar molecules that contain high energy electrons. Those electrons are transferred to NADH, which is also transferring high energy electrons. And then as the electrons are donated here, the energy that was present in the electrons is converted to an asymmetric distribution of hydrogen ions. So the energy is stored at this point in the concentration differences of protons across this membrane. And then finally, the energy is stored in the molecule of ATP as the proton gradient is relaxed, or protons go down the gradient. And so the energy that was present in the sugar is now present in the ATP molecule after being contained in the proton gradient, after being contained in high energy electrons. On this last slide for this video, I want to just introduce the 3 protein complexes. One that you should remember is the NADH dehydrogenase complex. Sorry, I'm not a better artist. That's relatively easy to remember. That's the highest energy complex, and you can always remember that because that's the protein complex which is going to accept the electrons from NADH, which is that small carrier. Another famous protein complex there that's a little better is Cytochrome C oxidase complex. I'll put the C there, that's sometimes there. And this is the final protein which is accepting electrons. It's donating it to oxygen. It's taking up protons from the solution. It's producing water. This is the protein complex which cyanide will bind to. And the way this prevents this process from occurring is cyanide binds to the site that oxygen normally would. However, cyanide binds more tightly, so oxygen does not gain access to the protein, and cyanide does not accept electrons. So the protein cannot put electrons on cyanide. It gets backed up. It gets backed up on these complexes. You can no longer pump protons across the membrane. You can no longer synthesize ATP. This cell has 2 minutes to live. The middle protein complex, you don't have to remember the name. Different people refer to it as different names. The book refers to it as Cytochrome BC1 complex. And I do want to introduce you to two intermediate electron carriers. We don't talk about them yet, or haven't talked about them much, because they don't do any proton pumping. All they're doing is they are intermediates and shuffling electrons between protein complexes. Yet they're both pretty interesting mechanistically. Ubiquinone is a small organic molecule. It's very hydrophobic. It is trapped forever inside the lipid bilayer. It will not go into the aqueous environment, but it's free to diffuse around in the membrane, and it is an electron carrier, so the NADH dehydrogenase complex can donate electrons to the ubiquinone, which can then further donate them to this protein complex. So this is a small organic molecule. Cytochrome C is a protein. It sits on the top of the membrane. The reason it won't diffuse away into the solution is because it has some fatty acids attached to it covalently, which acts like a string on a balloon. It tethers the protein to the surface because these fatty acids have to stay in the lipid bilayer. Like ubiquinone, cytochrome C can diffuse laterally inside the membrane attached to the hydrophobic tails, and it can work as an electron shuttle between complex 2 and complex 3. So two very interesting ways of transporting electrons, although these two molecules do not do chemistry in themselves. Okay, in the next video we'll see some of the mechanisms associated with proton pumping, and thanks for listening.